Up to now, we've talked about what enzymes are and how they bind substrates. Now let's answer the real question. How do enzymes actually make reactions happen faster? In this video, we'll review the basics of enzyme catalysis and the 4 main ways enzymes lower activation energy. To catalyze a reaction means to make the reaction happen faster without being permanently used up. A substance that does this is called a catalyst, and enzymes are biological catalysts made of protein. Enzymes do not change what the overall reaction produces, and they do not change the overall energy of the reaction. They only make it easier for the reaction to get started. Enzymes follow a simple sequence. The enzyme E binds to the substrate S, the molecule that the enzyme acts on, to form the enzyme substrate complex ES. The reaction occurs and the enzyme releases the product P. So E plus S goes to ES goes to E plus P. The enzyme is unchanged, so it can be used again. So you may have heard that enzymes lower the activation energy, but what does that actually mean? Let's take a look at the energy diagram drawn. The y-axis represents energy and the x-axis represents reaction progress. The red curve shows the reaction without an enzyme, and the green curve shows the reaction with the enzyme present. To react, molecules must reach a high energy state called the transition state. You can see that would be represented by like the top of the peak on both the hills. The energy needed to reach this point is called activation energy. Without an enzyme, the barrier is high, so the reaction happens slowly. With an enzyme, however, the activation energy is lower, so the reaction happens faster. Notice that the starting and ending energy are the same for both curves. Enzymes, again, do not change the overall energy of the reaction, and they do not change the equilibrium. They only lower the activation energy. You'll also see the ES label here for the enzyme substrate complex. It doesn't necessarily have to be at the peak, but it represents the bound state as the reaction moves towards the transition state. Most of the enzyme's catalytic work happens after the enzyme binds to the substrate, so inside the enzyme substrate complex. So the question becomes, how does the ES complex lower activation energy? And there are actually 4 main ways this happens. First, we have transition state stabilization. The transition state is the highest energy point of the reaction. Enzymes lower activation energy by binding the transition state more tightly than the normal substrate. And this stabilizes the high energy form and makes it easier to reach transition state. Second, there's microenvironment effects. The active site can create conditions that make the reaction easier. The enzyme may shield water, position charged amino acids, or create acidic or basic conditions, and these changes help the reaction occur more easily. Third, we have proximity and orientation. Reactions require molecules to collide in the correct position, and in solution, collisions are random. Inside the active site, the enzyme can hold the substrate in the exact orientation needed so bonds can break or form faster. Fourth, we have strain or distortion. The enzymes may bend the substrate toward the shape of the transition state, and this puts stress on certain bonds, making them easier to break. Because the substrate is already closer to the transition state, less energy is required. So these 4 ways are not separate steps. Different enzymes use different combinations of these strategies, but mostly rely on transition state stabilization plus maybe 1 or 2 others. Let's do a review of today's video. Enzymes catalyze reactions, meaning they make reactions faster without being used up. They follow the basic sequence of E plus S, it goes to ES, goes to E plus P. They speed up reactions by lowering activation energy, not changing the overall energy. And they do so by using transition state stabilization, microenvironment effects, proximity and orientation, or strain and distortion. And that is the basic mechanism of enzyme catalysis. In a future video, we'll look at enzyme kinetics, but that's kind of its own topic and I don't want to overload in one video. If this helped you at all, like, subscribe, leave a comment, share with a friend, and I'll see you in the next video. Thanks.